don't stop. Don't stop.
The nervous system is divided into the peripheral nervous system, PNs, and the central nervous system, CNS. The PNs is composed of 12 pairs of cranial nerves and 31 pairs of spinal nerves. The CNS is composed of the brain and spinal cord. The nervous system is a complex organization of tissue, ramifying throughout the body, functioning to collect information from within and from outside the environment of the body. The nervous system sorts this information, then reacts to perceived challenges imposed on it. Included within the functions of this system is the process of cognitive faculties occurring in the highest centers of the brain. The nervous system is divided morphologically, for descriptive purposes, into two divisions, the peripheral nervous system, PNs, and the central nervous system, CNS. The PNs comprises 12 pairs of cranial nerves emanating from the brain and 31 pairs of spinal nerves originating from the spinal cord. The CNS is composed of the brain and the spinal cord structure. The nervous system is made up of neurons, impulse-conducting cells, and neuroglial cells that support the neurons. The tissue of this system is made up of 1. Specialized cells, neurons, which conduct impulses. 2. And neuroglial cells, which serve in a supporting capacity. A neuron, the basic structural unit of the system, possesses the ability to perceive stimuli, irritability, and to transmit physiochemical impulses along its processes, conduction, to effector organs and/or other neurons. The neuron is composed of a cell body, soma, perikaryon, and its processes. 1. The cell body contains the nucleus and cytoplasm containing nacelle bodies, rough endoplasmic reticulum, which may be made visible by light microscopy through the use of special stains. Neuron cell bodies are located either in a ganglion, if outside the CNS, or in nuclei, if within the CNS. 2. Radiating from the cell body are freely branching processes called dendrites, which transmit impulses toward the cell body, and a singular axon, which transmits impulses away from the perikaryon. The axon emanates from the cell body at a bulge called the axon hillock. The arrangement of processes around the cell body might vary from that described here and are thus supplied with descriptive terms. Axons may or may not possess a fatty myelin sheath covering composed of the wrapping of Schwann cell plasmalemma, in the PNs, or oligodendroglial cell membrane, in the CNS. These myelin sheaths are of variable thickness and completely surround the axis cylinder, except at regular intervals along the axon. These intervals, called the nodes of Ranvier, impart a one-ink sausage appearance to the axon. Myelinated or unmyelinated axons are covered by a neurolemma sheath composed of Schwann cells, in the PNs, or oligodendroglia, in the CNS. The speed of conduction of an impulse along a nerve fiber is related to the absence or presence and the thickness of the myelin sheath. Functional components. Neurons are either of a sensory, afferent, function, an intercalated, connecting, function, or a motor, efferent, function. Neurons are categorized functionally as sensory, intercalated, connecting, or motor. Sensory, afferent, and motor, efferent, functions are each further subcategorized to facilitate description of a neuron's specific function. General somatic afferent, refers to sensory function, modality, perceived from the body and transmitted to the spinal cord or brain. Sensations such as pain, temperature, and touch to the skin are perceived by neurons in this category. Also in this category is sensation from muscles, tendons, and joints, referred to as proprioception. General visceral afferent, is the sensory modality received from within the viscera, glands, organs, and membranes. General somatic efferent, a motor component, serves to provide innervation to all of the skeletal muscles of somatic origin, whereas general visceral efferent stimulation provides motor innervation to smooth muscles, cardiac muscles, and glands. Sensory and motor functions within the head are carried by the cranial nerves. Certain muscle groups and the sense organs for hearing, smell, taste, and sight make this group special. The senses of sight and hearing are transmitted by special somatic afferent sensory neurons, whereas taste and smell are transmitted by special visceral afferent sensory fibers. Similarly, the motor component to the special muscles, branchiomeric origin, is the special visceral efferent. Because no special category exists for glandular secretomotor function in the head, the general visceral efferent component remains for the glands, smooth muscles, and mucous membranes of this region. Note that certain of the cranial nerves carry general visceral afferent sensory components from the viscera of the head as well. Peripheral nervous system, the PNs represents those nerve fibers that receive stimuli from the interior or exterior of the body, i.e., sight and hearing, and transmit it to the CNS for processing. The PNs also delivers motor function from the CNS to the periphery. Sensory neurons originating peripherally, example in skin, may possess nerve endings specialized for receiving various stimuli, such as cold, hot, touch, and pressure. Nerve endings transmitting pain, on the other hand, are free and without specializations. Dendrites of the spinal nerves are connected to their cell bodies located in the dorsal root ganglion, which is just outside the spinal cord. The axons pass from the ganglion via the dorsal root into the dorsal horn, sensory, of the spinal cord. Here they may terminate, enter the white matter of the cord to ascend or descend before synapsing on connecting neurons in the spinal cord, or ascend to conscious levels in the brain. Cell bodies of spinal motor neurons are located in the ventral horn, motor, of the spinal cord. Their axons traverse the ventral root on leaving the spinal cord. Just beyond the dorsal root ganglion area, the sensory and motor roots unite, forming a spinal nerve that thus carries both sensory and motor components. Motor fibers destined for muscle will continue on to synapse at the motor end plate, a specialized ending between the nerve and the muscle. Sensory nerve endings located, for example, in the patellar ligament of the knee do not have their terminals in the dorsal horn but, instead, terminate directly on motor neurons in the ventral horn of the spinal cord, thus affecting a reflex arc that bypasses connecting neurons. Rapid opening of the mouth as a result of painful stimuli from biting down on a piece of bone while chewing is an example of a reflex arc in the fifth cranial nerve. Central nervous system. The central nervous system is composed of the brain and spinal cord. The central nervous system is represented by the brain, which is housed with the skull, and the spinal cord, which is housed within the vertebral canal surrounded by the divisions of the vertebrae. The brain and spinal cord are responsible for analysis, integration, and response for the body via sensory input and motor output. Coverings. The CNS possesses three separate meninges that encase it: dura mater, arachnoid, and the pia mater. The CNS is composed of the brain and the spinal cord. Each is delicately covered by several layers of meninges and is protected by bone, either the skull around the brain or the bony vertebral column that surrounds the spinal cord. The meninges covering the brain and spinal cord are continuous and completely enclose the CNS. Three separate layers make up the meninges: a tough outer layer, the dura mater, an inner delicate layer closely applied to the brain and the spinal cord and their vessels, the pia mater, and an intermediate layer, the arachnoid, which is closely applied to the dura. Only a potential space exists between the dura and arachnoid, known as the subdural space. The subarachnoid space, located between the arachnoid and pia layers, contains the cerebrospinal fluid that functions in bathing and further protecting the CNS. Autonomic system. The autonomic nervous system, by definition, is a motor system controlling the viscera, cardiac and smooth muscle, and glands. It is subdivided into the enteric, sympathetic, and parasympathetic systems. The autonomic, involuntary, visceral, nervous system exerts control over the viscera of the body, serving cardiac muscle, smooth muscle, and or glands. By definition, the autonomic nervous system is purely motor in function. Its manner of functioning is different from that previously described in that innervation is accomplished via a two-neuron chain between the CNS and the effector organ, smooth muscle, cardiac muscle, and glands. The cell body of the first neuron in the chain is located in the CNS, brain or spinal cord, whereas the cell body of the second neuron is located in one of the autonomic ganglia, all of which lie outside the CNS. The autonomic system is subdivided into 1. The enteric, 2. Sympathetic, 3. Parasympathetic systems. The enteric nervous system is located in the wall of the digestive system and functions in the autonomic control of the digestive system. It is influenced by the other two components of the autonomic system. The enteric nervous system is not associated with the head and neck and, therefore, will not be discussed in this textbook. Sympathetic nervous system. The sympathetic nervous system is that system which puts the body ready for action. The sympathetic system generally prepares the body for action, as in the fight or flight response, by increasing heart rate, respiration, blood pressure, and blood flow to the skeletal muscles, dilating the pupils, and generally shutting down visceral activity. Neurons of the sympathetic system originate in the intermediolateral cell column of the spinal cord in the thoracic and upper lumbar segments, Ti2L2L3. Thus, they are often referred to as the thoracolumbar outflow of visceral efferent fibers. Parasympathetic nervous system. The parasympathetic nervous system serves to calm the body, returning it to a homeostatic state. Parasympathetic innervation, conversely, functions to calm the body by decreasing heart rate, respiration, and blood pressure, constricting the pupils, and increasing visceral activity. Both systems innervate many organs of the body where their antagonistic actions serve to balance functioning to maintain homeostasis. Neurons of the parasympathetic system originate either in the brain in certain nuclei of cranial nerves 3, 7, 9, and X, cranial outflow, or in the sacral spinal cord from the intermediolateral cell column of spinal nerves S2, S4, sacral outflow. Together, this system is known as the craniosacral outflow. The cell body of the first neuron within the two-neuron chain of the autonomic system is located within the visceral efferent column of the CNS. The axon of this neuron will synapse on the cell body of the second neuron in the chain, located in one of the autonomic ganglia. Thus, this axon is preganglionic. The axon of the second neuron is postganglionic and extends to the effector organ. The sympathetic system is served by the autonomic ganglia located along most of the spinal segments. These ganglia are known as the sympathetic chain ganglia, paravertebral ganglia, and are connected to each other by the sympathetic trunk and the several collateral ganglia, preaortic ganglia along the major abdominal blood vessels. Ganglia of the parasympathetic system are located close to the structures innervat and are called terminal ganglia, 4 of which are in the head, whereas others, the enteric ganglia, are located within the wall of the alimentary canal. Preganglionic sympathetic fibers reach the chain ganglia via the white rami communicantes, a connection between the spinal nerve and the ganglion transmitting the myelinated fibers. The postganglionic fiber may enter the spinal nerve via the gray rami communicantes directly or after ascending or descending in the sympathetic trunk. Preganglionic fibers synapse only one time, therefore, those destined to synapse in the collateral ganglia do not synapse in the chain ganglia. Preganglionic parasympathetic neurons of the cranial outflow originate in cranial nerves 3, 7, 9, and X only and may be distributed to the terminal ganglion via the cranial nerve of origin or by the named preganglionic fiber. Postganglionic fibers are distributed by other nerves serving the organ. Acetylcholine is the neurotransmitter of both preganglionic sympathetic and parasympathetic neurons and postganglionic parasympathetic neurons. However, noradrenaline is the primary neurotransmitter of postganglionic sympathetic neurons except those serving sweat glands. In this case, the neurotransmitter is acetylcholine. The suprarenal, adrenal, medulla functions as a modified sympathetic ganglion. It receives preganglionic sympathetic fibers which synapse on chromaffin cells of the medulla, stimulating them to produce catecholamines, which give rise to epinephrine and norepinephrine, which are released into the bloodstream. The epinephrine produced here prepares the body for fight or flight during stress or fear. The norepinephrine produced here is short-lived.